Good afternoon, everybody. First, I would like to thank you all for the honor of this masterclass. It's a great honor for me to give the masterclass here in Bologna, our hometown and home university. And I'm happy to tell you something about the multidimensional complexity of opioid receptor in the frame of developing innovative and possibly improved therapeutics. Here is the presentation outline. I will start by summarizing what is the current evolving view of GPCR pharmacology and how to experimentally study it, investigate it in the frame of developing improved therapeutics. Then I will touch briefly upon some background information on opioid receptors and ligands, and this way, later on, we will be able to discuss together about promises and pitfalls of developing more effective and safer analgesics targeting the opioid receptor, and also to talk together about opioid receptor that is emerging as another potentially interesting therapeutic target. And of course, last but not the least, I would also like to conclude with some aspects related to quantitative system pharmacology based approach. Again, still in the frame of developing improved therapeutics and considering the multidimensional complexity of opioid receptor. G-protein-coupled receptor for decades have been believed to determine their effect exclusively by activating and modulating the function of the G-protein, and hence their name, G-protein-coupled receptor. Now we know that their signaling pathways and modalities are way more complex, and we can have G-protein-dependent signaling components, arrest-independent signaling components, and interestingly, depending on the ligands engaging the receptor, one subset of the signaling pathways can be preferentially activated. And moreover, you can have receptor internalization, but different ligands all internalizing the receptor may determine arrestin-mediated recruitment of distinct subset of effectors, scaffold proteins, thus further differentially shaping the responses that are elicited downstream. And also, it has been reported in literature that the second and third waves of both G-protein and arrestin-mediated signaling may occur later on with spatial and temporal bias. Classically, ligands and GPCR have been described in terms of their activity based on the concept of intrinsic efficacy. So you have full agonist here, an agonist that binds to the receptor and fully activate all the signaling pathways linked to that specific receptor to the maximal extent. Then you may have partial agonist activating all the signaling pathways at submaximal effect, and then antagonist or inverse agonist. And this has shaped drug discovery programs for a while. But an accumulating body of evidence has been accumulating in the past years, and now it's clear that different agonists at the same receptor may differentially modulate downstream signaling pathways. And here, in the example provided in the slide, you can see agonist A, for instance, more potent and more efficacious in triggering signaling pathway A over B as compared to agonist B. So bottom line is that different agonists at the same G-protein-coupled receptor do not activate all the responses to the same extent. This can be interesting also in the therapeutic perspective. And all these findings that in past years and decades were mainly dismissed either as errors or as specific events occurring in very distinct and narrow experimental settings were later on accepted as a phenomenon, functional selectivity. And I like this paper quite a lot. It was published in 2007. Because to me, it represents one of the first papers in which this phenomenon has been clearly stated, described, also showing that it was accepted by the scientific community. If functional selectivity is a real thing, this opens to further interesting questions. First, how mechanistically is possible that different ligands at the same receptor triggers different effects in terms of activation of intracellular signaling? Second, which effects are then induced at the level of target tissues, organs, and organism? And ultimately, which can be the physiological and more relevantly the pharmacological consequences of this functional selectivity? Can we exploit these phenomena in a therapeutic perspective? And to experimentally study this aspect, first in vitro, it is important to employ different cell models with both heterologous and endogenous expression of the target receptor, because if you just focus on one cellular background, you can capture some effect that is system dependent, but it's not related to a real effect that is translatable to other systems. Also, it's important to always include the proper reference balance agonist, meaning the ligand known to bind that receptor and activate all the signaling pathways as a reference compound, and possibly to exploit more than one assay or experimental readout for both G protein and arrest-independent component on intracellular signaling. Also, how to quantify this biased agonism has been debated, and the proper quantification can be relevant to guide, for instance, structural activity studies, and also for the comparison of different ligands and guiding the selection of candidates that may be more successful later on in the development process. But it's important to realize that G protein versus arrestin is just the first branching point in functional selectivity. And it would be important also to focus on more highly complex patterns of functional selectivity downstream of G protein and arrestin. And for instance, it can be useful to focus on differential activation of signal transducers, for instance, MAC kinases, differential modulation of gene expression, ligand-dependent modulation of whole cell responses, such as proliferation, apoptosis, or depending on the therapeutic target topic. Another caveat that is important to be reminded of is that most of the studies within this functional selectivity have been carried out in vitro and exploiting the heterologous expression system. This is useful, but it may lead to miss some important component in the whole picture. And so it would be crucial to transfer this kind of studies to primary cultures, tissue preparation, and in vivo interclinical model. This is interesting because if you administer to a rodent, to a mouse, for instance, two different agonists, the orange and the blue one, you may observe differential modulation of a given response. But functional selectivity is not the only mechanistic explanation for these differences, because, for instance, it's clear you see the orange agonist fully activated in both response 1 and 2, and the blue guy activating on the response 2. So you can say, okay, it's functional selectivity, it's biased agonism. That can be true because the two agonists may differentially modulate the target receptor, but there are a bunch of other very reasonable possible explanations. For instance, in vivo, the blue agonist may bind to another G protein coupled receptor belonging to a different family, and the result of this other activation counteracts the activation of response 1 downstream of the first receptor. And in the end, you have again the differential modulation of the final response, but the mechanism is different. Or in vivo, the light blue agonist can be metabolized into an antagonist to the same receptor. And again, the combination leads to the differential modulation of the desired response, but the molecular determinants do not depend on biased agonism. Or, this is also interesting to me, that two different responses can be constrained into different compartments within the human body, for instance, the brain and the periphery. And so the differential response may be just due to different pharmacokinetics. More simple explanation. And all this needs to be dug up and dissected in order not to be fooled by an hypothesis that it's likely to be confirmed, but it's not. As you know, endogenous opioid system includes 4 receptors, the classical nu-delta kappa and nociceptive receptor, and their related endogenous peptides, each displaying specific affinity and selectivity for the target receptors. Opioid receptors are class GA, GPCR, and they are involved in the modulation of several physiological and pathological processes, not limited to pain. They are important in pain, but also in mood regulation, immune function modulation, feeding behaviors. They are typically coupled to G-alpha proteins, and so upon the activation by agonist, adenyl cyclase is inhibited due to the activation of G-alpha. The beta-gamma subunit inhibits presynaptic calcium channel and activates postsynaptic potassium channels. All these signaling events leading to analgesia, but also MAP kinases can be activated in a G protein-dependent fashion. Receptor can undergo phosphorylation, and this is also different depending on the agonist. Arrestin can be recruited and trigger further signaling events, can modulate receptor trafficking, desensitization, and the link to tolerance. From a pharmacological point of view, the more receptor has been the most relevant so far, the most exploited in terms of therapeutics, as it is the target of the opioid analgesics currently used in clinic, like morphine, fentanyl, oxycodone, and the other. And as mentioned, its activation by agonists triggers multiple signaling pathways, ultimately leading to the desired effect, analgesia, but also to relevant side effects, constipation, nausea, vomiting, respiratory depression, which is of relevance, immunomodulation, tolerance, addiction. And despite these relevant side effects and the issue of abuse liability, as you know, analgesics are still among the most powerful and widely used painkillers for moderate to severe pain, acute, but also for chronic pain. Nonetheless, we know very well that good majority of chronic pain patients treated with this kind of drugs and other, they do report either inadequate analgesia or unbearable side effects or both. There is the issue of addiction and abuse liability, and it's clear, otherwise we won't be here as a consortium, that it's very urgent, the need to develop more effective and safer analgesics. And indeed, this attracted efforts for decades. And despite that, if you think that we are still using morphine, whose analgesic and also euphoric properties were known thousands of years before Christ, one could think that we did not progress at all. It's not fully progressed, but not enough. And in fact, some authors, in a provocatory way, they compare these efforts to the quest for the Holy Grail, highlighting that it's very hard to get there. Is it possible to get there? Will we ever make there? And there are many different strategies that have been undertaken. I listed here most frequently used, or the one you may most frequently find in literature. And I will comment the three highlighted in both: so biased agonism, low intrinsic efficacy agonist, and combination treatment. That is something that we, as a consortium, like very much. Biased agonism can be defined as the ability of the ligand, the G protein couplet receptor, to trigger distinct signaling pathway downstream of the receptor over others. And this is due to the stabilization of active conformation of the receptor. If you compare different agonists, even full agonists, they can stabilize a slightly different conformation. And this influences the subset of intracellular pathways that are triggered as a consequence. And if you think at this phenomenon in a therapeutic perspective, and if we understand which pathways are more related to the therapeutic effect and which are responsible for the adverse effect, this may represent an intriguing opportunity if we find a ligand triggering mainly the blue effects in this slide, like here, selective agonist A, we are likely to have in our hands a molecule with improved pharmacological profile. And these hypotheses and these findings arose a great interest in the opioid research field in the past decade, 10, 15 years. And we will come back to it later. But differential modulation of GPCRs can be obtained also exploiting different intrinsic efficacy of ligands. If you look at this graph here, 4 agonists at this receptor are depicted. They all display the same potency because EC50 values are overlapping, but they clearly display much different efficacy. And so, for instance, agonist A, the red one, high efficacy, is likely to activate both pathways to a high extent. But B, lower intrinsic efficacy agonist, may activate the receptor to a submaximal extent that is enough to trigger pathway 1, but it's not enough to trigger in a significant way pathway B. And this is also a way to reach functionally selective modulation of G protein couplet receptor. And of course, as we know very well, it's the aim of our consortium identify combinational treatments. And if you think about agonists at different GPCRs that are properly selected and tailored, the overall effects elicited may provide a more balanced profile in terms of therapeutic desired effect versus unwanted side effects. Going back to bias agonist and new opioid receptor, this attracted, as I mentioned, a great interest and effort in the past 10-15 years. And indeed, it's an hypothesis-driven strategy because 20-25 years ago or so, it was demonstrated in arrestin 3 knockout mice that morphine displayed improved pharmacological profile in terms of enhanced analgesia, reduced tolerance, and fewer adverse effects. And similar results were recapitulated in rats, in which arrestin expression was knocked down via administration of siRNAs. On the other hand, all over the past years and decades, quite a huge number of new agonists have been developed and studied, and they differ for structure, features, and properties, but in the end, they share nearly the same set of adverse effect as morphine. And so it became clear that both analgesic and unwanted effects were related to the activation of opioid receptor. Within this frame, the possibility of connecting also to the data showing the improved profile in arrestin 3 knockout mice or arrestin 3 knockdown rats, the possibility to triggering preferentially the signaling pathways leading to analgesia or limiting the activation of the signaling pathways leading to adverse effect appeared really promising. And here it's a picture summarizing the working hypothesis here. And within this frame, I think it's very paradigmatic and education on the story of TRV 130. Oriceridine is the name of the compound. It's been developed by Trevina, and this is a paper from 11 years ago. And here, authors show that the TRV 130, this molecule here, was able to activate G-protein-dependent signaling downstream of opioid receptor, although you may see it's not full agonism, with negligible recruitment and activation of arrestin compared to morphine that activates both branches of signaling pathway downstream of opioid receptor. Very interesting profile. They move forward to in vivo assays in rodents, and here you see TRV in black eliciting antinociception, and in orange and blue, it's two different measures of gastrointestinal adverse effect that were observed at concentration higher than the analgesic one. So a sort of separation between wanted and unwanted effect, whereas morphine triggers both desired and undesired effects in the same concentration range, or even adverse intestinal adverse effect appears at lower concentration. It's similar with the respiratory adverse effects. And so from there, they moved forward and rather quickly to clinical study. And in healthy volunteers, TRV 130 was effective in producing antinociception with less reduction in respiratory drive and with fewer gastrointestinal adverse effects as compared to morphine. Although the separation, if we look at the graph here, the separation between the curves of wanted and unwanted effect was lower than in rodents, and this should have sounded as a warning signal before going forward. Then they moved to a phase 2 clinical trial or some phase 2 clinical trials, and indeed the analgesic effect was greater as compared to morphine, but no any improvement in terms of the balance with respect to adverse effect. This is kind of interesting. So it seems that from in vitro to preclinical to clinical, the improved pharmacological profile will be narrow. They, in early 2018, announced the filing of FDA application, and they call it the first receptor G-protein pathway selective modulator. But in October 2018, the FDA committee voted against approval. It was a tight vote, to be honest. 8 were against, 7 were in favor. So it means that it was quite a debate. But in the end, it was not approved as superior as compared to morphine. And this, I remember very well, because at that time we were also very interested in biased opioid receptor agonists, and the enthusiasm really tamped down in the community because it was felt that probably the hypothesis was not good enough, or important things were missed, or some authors started doubting that functional selectivity is something real and exploitable. And in fact, it emerged that the molecule retained undesirable effects that were G-protein determined, some abuse liability, but after further phase 3 clinical studies, it was approved in 2020 for the treatment of postoperative pain, intravenous administration, but as non-superior as compared to morphine. So it's available in the US as an alternative to classic opioid agonist. So it's not bad, but it's not the improved pharmacological profile that researchers aimed for from the beginning. And it also turned out, as I already anticipated, that some adverse effects initially believed to be arrest independent, they do also depend on some G protein component in the signaling. So it's not that the good guys are on one side and the bad guys are all on the other side. It's more complex. This is also kind of interesting because knocking mice strain was generated expressing a G protein biased more, meaning a opioid receptor that cannot recruit arrestin. And you would expect in these mice to reproduce an improved pharmacological profile if you administer morphine. This was not the case. Again, supporting that finding that it's much more complicated. And some other authors started repeating the original experiment from 2000, and they couldn't reproduce them. And this led also to doubt about those experiments, but I believe, and the belief is that it's a matter of the strain of mice that was used, that it's not the same that is used here, meaning that there are system-dependent components that affected the generation of the hypothesis. And so, but the hypothesis has been challenged. And in this other paper, it is interesting because authors, and these are skeptic about bias, they selected 5 different readouts for G protein activation and 3 different readouts for arrestin activation. And they tested Tango as a reference compound and different opioid drugs currently used in clinic, as you see here, and 3 also buprenorphine, and 3 they call putative bias agonists because these authors do not believe in that much in functional selectivity due to bias. They in vitro quantify all the pharmacological parameters for potency, efficacy, intrinsic efficacy. Then they move to rodents and they compare antinociceptive effect. It's the darker colored line in each graph on the left, and respiratory adverse effect is the lighter colored curve on the right side of each graph. And in the between, there is the therapeutic windows. And of course, as we know, fentanyl is quite the narrow therapeutic windows. Buprenorphine has wider therapeutic windows. And then they try to correlate the therapeutic window to bias factor, no any correlation, to intrinsic efficacy, inverse correlation. Buprenorphine in their system, in their setting and analysis, is advantageous because it's low intrinsic efficacy and nothing to deal with the modulation of distinct subset of signaling pathways. And this started in a field, the fight between opposing parties, those supporting one or the other idea. And personally, I don't like this kind of dynamic because I think it's detrimental for the field. In the end, it's important that we produce knowledge that can be used for the benefit of patients. And if in the end it's one or the other, the way that will get us to improve analgesics, who cares? It's important to get there. But the field got a bit polluted, so to say. So we are still here with the Hamletic conundrum. So is it bias or is not bias? I believe that we cancel out that functional selective bias can be exploited. Low intrinsic efficacy can be also a smart strategy. And I think that the most important lesson to be learned is that every hypothesis needs to be challenged, verified, investigated in with multiple strategies and complementary at different levels, because something can be reasonable, promising, but you need to be sure also in the translation. And if I think back at the TRV trajectory, when they saw that the separation between wanted and unwanted effect was getting narrower, it should sound as an alarm to say, okay, let's wait a bit, focus more on what can be going on there. Moving to the kappa opioid receptor, which is at the moment my favorite opioid receptor beside the blue. It's interesting. It is contributing to a variety of physiological processes, nociception, stress, mood regulation, gap motility, and core agonists. They need have been explored as alternatives to more analgesic because they do not have abuse liability. They have the opposite problem. And typically they induce lower respiratory and gastrointestinal adverse effects. Unfortunately, so far, no any core agonist can be used in clinic due to severe central adverse effects, sedation, but more importantly, dysphoria. That's why they do not have abuse liability. They are the opposite. They induce dysphoria. Interestingly, also downstream of kappa receptor, it's been shown that part of the wanted effect depend on G protein activation, and some, not all, but some of the adverse effect, sedation for sure, motor incoordination for sure, some component of the dysphoria, some anhedonia is arrest independent. And so why not trying to develop G protein bias core agonist? This is quite attractive at the moment in the opioid field. Moreover, it's been shown that the dysregulation or overactivation of endogenous core system in specific brain areas by the endogenous peptide dynorphin contributes significantly to the craving for substance of abuse in some phases of addiction, also to persistent symptoms typical of depressive disorders, and also to some extent to the positive symptoms of schizophrenia. Therefore, there is a growing interest in core antagonists to be used for the treatment of this psychiatric condition. And to no surprise, recently, aticaprant, a core-selective short-acting antagonist, reached the clinical trial to test its effect in combination with SSRI as compared to the SSRI alone. And with respect to different readout, the combination, the green curves here, are better than the SSRI alone. And thinking about pain and comorbid depression, which are tightly connected, because chronic pain, we know it's related to different biological, social, and psychological factors. Also, the negative affective state may influence long-term consequences of chronic pain, and the other way around. Premorbid psychological dysfunction may predispose subject to develop chronic pain later on. So there is this bidirectional connection. And thinking that core activation in pain pathways may lead to abuse liability free analgesia, and counteracting core dysregulation may lead to antidepressive effect. This poses the kappa receptor at the crossroad between these comorbidities that can be possibly exploited to develop innovative agonists as promising improved therapeutics. And again, it's important to be reminded of the fact that some of the wanted effects are G protein dependent, adverse effects, some of them are arrest independent, and also some of the core-mediated effects leading to depression are in part due to arrest inactivation. So one could hypothesize that the ligand activating this pathway can be interesting to treat the pain, but also to limit, at least in part, some core-mediated components of depression. And within this frame, our research group have been dedicated for years to the characterization of innovative opioid peptides to detail their functional selectivity and if this functional selectivity can be connected to an improved pharmacological profile. And so we employed multiple cell models, both heterologous and endogenous expression. We tested multiple readouts, arresting G protein, downstream higher level functional selective readouts, whole cell responses, and in vivo, different antinociceptive and pain model, and different tests for some side effects or adverse effects, because in order to have as much information as possible. And with this frame, there are a couple of ligands I would like to tell you something about. They were developed in collaboration with Professor Gentilucci Group from University of Bologna. And here you have LOR17. It's a cyclopeptide, highly selective and with high affinity for the kappa receptor, able to inhibit adenylyl cyclase in 3 different cell models with potency and efficacy comparable to the reference balanced core agonist, U50, that we use as a reference. And looking at some graph, here you see G protein-dependent signaling. The two are determining the same effect, but LOR17 displayed negligible arrestin activation as compared to the core reference ligand. And this was kind of interesting. And we then quantified the parameters, the same parameters that in that paper, the 2020 paper, were calculated by the authors that were skeptic about biased agonism. And we found that LOR17 indeed is quite high efficacy. And so that's the reason why you have maximal efficacy and potency overlapping to the reference agonist. And so indeed here, the determinant for the differential response is biased agonism. We move to downstream higher level functional selective readouts. And it's known that earlier captivation downstream of kappa receptor is protein dependent. Later, and P38 activation is arrestin-free. So we very simply studied these phosphorylation levels via Western blot. So in principle, quite an easy basic experiment. Interestingly, we found out that U50 in black activated in all the 3 cell models the early and the late phase of phosphorylation, G protein dependent, arrestin dependent. LOR17 only the first wave that is G protein dependent, and consistently LOR17 did not activate P38 in neither of the cell model, but U50 did so as expected. We move to whole cell response, and we choose astrocyte proliferation because it's a measure of astrocyte activation, and glial contribution to neuroinflammation in pain is well known. So this was our feeling a good cellular response to investigate. And we demonstrated that U50 can increase astrocyte cell proliferation. This is core dependent because it is reversed by the core antagonist. Is arrestin dependent because you don't have the effect in cells whose arrestin-free expression is knocked down. Is P38 dependent because it's sensitive to the P38 selective inhibitor. LOR17 does not trigger it. And higher concentration of LOR17 prevents U50 from activating it. The same in primary cultures of human astrocytes. So LOR17 does not activate it, not even at counter micromolar. So this confirmed the robust pharmacological profile in vitro. We move to in vivo, the warm water withdrawal test. So the basal latency is measured and animals are treated. Latency to response is measured at different time points. This is antinociceptive test. And we found that both ligands determine a dose-dependent antinociception with very similar potency and quite similar, or not too different, time-dependent profile. So again, quite good. And then thanks to the collaboration with the Professor Cesare Mannelli, University of Florence, they have a very translationable model of oxaliplatin-induced neuropathic pain. So mice are treated for 2 weeks with oxaliplatin, and the doses range resemble that is used in human to treat cancer. They develop neuropathy, and then you treat with the vehicle or the compound under investigation. Here are the healthy animal. Here is thermal hyperalgesia in the neuropathic animal, and LOR17 was highly effective in counteracting thermal hyperalgesia, much better than U50. And if you look at the side effects, here, number of falls in the rotarod, so it's a measure of motoring coordination. LOR17 and vehicle are the same, whereas U50 induce motoring coordination as expected. And mobility time in the forced swim test, it's kind of a measurement for prodepressant-like effects. Again, U50 is prodepressant, LOR17 is exactly as the vehicle. So the improved pharmacological profile that we observed in vitro was recapitulated to some extent in vivo, and this we think it's a good basis to further develop the compound. Another interesting to us compound is CL39. It was identified in a small library of endomorphin 1 analogs in which the second amino acid proline was replaced by this group in all the 4 possible configurations. And again, CL39, highly selective, high affinity to the kappa receptor, partial agonist behavior that was confirmed in all the three cell models. So the experimental setting was the same as for ROR17, and it's clearly CL39 in blue, partial agonist, always the reference agonist was tested in parallel. Interestingly, CL39 did not significantly activate arrest-independent signaling. This was checked with two different experimental settings. At this time, yes, likely not biased, but low intrinsic efficacy. And out of the concentration response curve, we measure the quantitative parameters, and indeed, CL39, it's a low intrinsic efficacy partial agonist because the maximal response is much lower than the full agonist, slightly more potent than the reference agonist. We tested also in vitro, and let's move to in vivo data for the sake of time. Antinociceptive in the water withdrawal test, partial agonist profile, core-dependent effect also in vivo because it is reversed when we co-administer the selective antagonist. And interestingly, again, much better in counteracting both thermal hyperalgesia and mechanical allodynia as compared to U50. This was, again, carried out in collaboration with the Florence group. And for CL39, we don't have this part for ROR17, thanks to collaboration with Dr. Brian Reed in that time at Rockefeller University in New York. We could test whether CL39 is aversive or not with the conditional place preference aversion paradigm. U50 is aversive as compared to vehicle. CL39, not that much. It's not exactly as the vehicle, but it's much less than U50. So again, interesting profile and quite enough matching between the in vitro and the in vivo plan. We would like, if possible, to further develop this compound. And the most recent one is this RDM1127. We identified it in a library of compounds derived from the core-selective G-protein biased agonist. Again, it's highly selective and high affinity to the kappa receptor, but it does not displace fully the available ligand from the orthosteric side, so suggestive of allosteric binding. Administered alone, it does not inhibit the recyclase, but interestingly, if we administer together with U50, the curves are shifted rightwards and upward, and this is indicative of negative allosteric modulation. Further studies will be performed in vitro to confirm on higher level readouts, and we would like to test it also in vivo, but the profile is compatible with negative allosteric modulation, and to our knowledge, this is the first ligand reported with this kind of profile. And going back to our starting point, chronic pain and comorbid depression, these three pharmacological profiles may all be relevant because with the G-protein biased core agonist, you can have analgesic effect, but you can limit at least part of the core-dependent component contributing to depression, not all the component. CL39, partial agonist, you may expect the effect as analgesic in chronic pain. In depression, it can limit the activation of some core-dependent prodepressant effect, and also partial agonist when there are high concentration of the full agonist behave as antagonist. So that could be also another interesting mechanism to exploit. A negative allosteric modulator, as core antagonists do, may be beneficial to treat depression, but also pain, because at least in preclinical model of indifferent preclinical model of chronic pain or neuropathic pain, it's been shown that core activation by endogenous dynorphin on glial cells significantly contribute to chronic pain maintenance. So it can be the scenario two birds with one stone, question mark, so a bit of wishful thinking, but I think we believe that, and yes, that this can be a smart way to try to modulate this complex scenario of opioid receptor to get to improve therapeutics. But the complexity is very big because we talked about G-protein and arrestin. There are isoforms that can be expressed differentially in different places in the body, different times. Also, physiological condition can impact on the effect. Receptor may dimerize, they are not isolated monads. Pain itself changes the system because one thing is antinociception, one thing is analgesia. And in pain, the system is changed dramatically and this impact. And so we need quantitative understanding of all the mechanism and also comprehensive, integrated, and network-centric approaches. And so, of course, this is quantitative systems pharmacology provides answer to. And here comes our project. And as you know, in QSPainRelief project, we are tasked with providing in vitro data to cover this part. And this has also something to do with the complexity of opioid receptor system. And so one thing that for us to be relevant here is the drug concentration to be used. And so the concentration were calculated by Bersin and University of Leiden, and this was important to us to test them on receptor expression because receptor density and expression can be a measure of density is relevant to the final effect. And so we could measure expression of the receptors. But also one other point that to me is important is receptor activability. We pretreated the cells, the same in every model. Here it's differentiated stage with a covalent antagonist to willingly reduce the number of receptors that can be activated, or we treated the cells for a prolonged time with morphine to induce tolerance-like state. And in both scenarios, morphine works in a worse way. It's not surprising, but what is a bit more surprising is that with the covalent antagonist, the number of receptors available to morphine binding is reduced. And it's clear that here, if you measure occupancy, you have a good link between occupancy and signaling readout. Here, you have the sensitization, but the number of receptors that can be bound are identical, are the same, but the receptor is not working. In here, if you use occupancy, you may be missing the fact that the receptor is not working. That's why I don't know how, but including in part activability could be beneficial to the platform. We are aware that we are only looking at cyclic MP, one of the components, and the system is way more complex, and you have also dimerization to take into consideration. But in the end, I think that in our platform, that hopefully will help identifying combination that may help different subset of patients, if not different individuals, will be able to include, at least in part, also the multidimensional complexity of opioid receptor. And these approaches, quantitative system pharmacology approach, could be also, in our view, applied to other fields of opioid receptor research, like biased agonists, because there are a lot of black boxes in all the process. And if we could shed at least some light at this level, the likelihood of getting really effective biased agonists could be increased. So going to the concluding remarks, biased agonists more, I believe, are still interesting, but it's the same for innovative ligands. Working hypothesis should be thoroughly validated. And the failure to translate all the intense endeavor of these last decades is connected to the highly complex multidimensional pharmacology of this target receptor. And within this frame, quantitative system pharmacology-based approach will for sure help improving the route to reaching the Holy Grail, hopefully, which in our case is not an ancient cup, but it's tackling down pain for our patient. And before concluding my presentation, if I may, I would like to dedicate this masterclass to Professor Spampinato, who unexpectedly and suddenly passed away nearly 1 year ago. As you know, he was a member of our research team, a leader for many years, and most of all, carrying on mentor for me and Monica Baiula. And we like to remember his strong and contagious passion for research, his relentless dedication to teaching and mentoring young generations of young researchers, including us, and the deepest respect for colleagues in the institution. And now it's our duty to bring forward these attitudes in our daily activity. And of course, a big thank you to my research group, to all the collaborators, to the research data I showed you today, our funding sources, and all of you for your attention, and happy to take any questions.